Hey everybody, in this video we are going to talk about epigenetics. Epigenetics. Okay, so what is epigenetics? So here's a nice definition. Heritable phenotype changes that do not include alterations in DNA sequence. Okay, so these are phenotype changes that do not include alterations in DNA sequence. You know, how can that happen, right? So far in this course we've been, discussing or looking at examples of how changes in the DNA sequence, changes in genotype, can lead or cause the change in phenotype. So now we have an instance, or we're going to look at some instances where the phenotype can change and it's independent of the DNA sequence. And the definition of epigenetics can be complicated because there are lots of examples where there are changes occurring and there are no in phenotype, but there is no change in DNA sequence, and then you have to think, well, is it heritable or is it not heritable? And there may be debate on whether or not the phenotype that we're looking at is heritable. So definitions of epigenetics can be confusing, and I guess we'll focus on this one, that to really be considered an epigenetic phenomenon, the phenotype must be heritable, in addition to not including an alteration in DNA sequence. So the phenotype change must be heritable, in addition to not including alterations in DNA sequence. So the example we're going to look at in this video is genomic imprinting. And in genomic imprinting, DNA is marked. DNA is marked, and the mark is retained throughout life of the organism. So the DNA is marked and the mark is retained throughout life of the organism. And this term imprinting, well, there are parallels, and maybe it even comes from this. So let's say these are baby birds here, and there are a lot of faculty here at ISU that study bird biology. I am not one of them, but we know from our notes that this term imprinting comes from, or there's an example where baby birds will imprint on their parents, and they'll look for certain marks, on their parents. And let's say this mom bird here, or dad bird, has some funny marks on it, and the birds will notice these marks, and that way it'll be able to see or know which individuals are its parents and which are not. At least that's how I think imprinting works in birds. Now, genomic imprinting, in the imprinting, it implies a sort of memory. So something's being remembered, and the baby birds are remembering the marks. In genomic imprinting, the thing that's being remembered is the marks on the DNA. They're being retained throughout the life of the organism. So what do we mean by marks? What do we mean? So sometimes marks might not occur directly on DNA, but there could be the histone code, right? Histone code involves what? We saw this in the last video, covalent modifications of histone tails. And maybe these specific modifications, maybe those can be maintained throughout the life of the organism. So that's one possibility. So DNA methylation, which we also talked about, I guess histone code may have been 2 videos ago. I forget. So DNA methylation, I believe. DNA methylation with respect to CpG islands. We talked about that in the last video. So those marks, the DNA methylation, this is a type of mark that can be retained throughout the life of the organism. Now we're going to look at an example of how DNA methylation can lead to a phenotype change. And the example we are going to look at involves the IGF-2 gene, which is stands for insulin-like growth factor 2 in mice. And there's a similar gene in humans, I'm sure. I haven't looked it up, but almost every gene in mouse, every gene in mouse is also found in humans. Not all, I'm sure, but most. So now, okay, let's look at mice, and we are going to talk about this gene. And instead of calling it IGF-2, I'm going to make things easy on myself, and I'm just going to label it I. Okay, so the I plus, the wild type allele, I'm going to label I plus. A mutant allele, I'll label I minus. And so I minus will be a null allele, so not functional. And something interesting happens here when we do crosses with mice with these 2 alleles. So let's say we have homozygous I minus for the mom here. And when a mouse is homozygous null like this, they have a dwarf phenotype. So here is a dad, homozygous wild type, has the normal phenotype. Now if we do this cross, the offspring, it doesn't matter, males or females, sons or daughters, whatever, they all have the same genotype, right? So they're getting an I minus from mom, an I plus from dad, and they're normal. None of them are dwarf. So, okay, well, that's not too surprising. Maybe I plus is dominant to I minus. Okay, so, I mean, we've seen this many, many times in the past. So how about if we do the reciprocal cross? So this is a term we saw in previous lectures. So the reciprocal cross is where we exchange the genotype with the sex. So here the mom was I minus, I minus. So over here, the mom is going to be I plus, I plus. So the mom has the normal phenotype, and the dad is a dwarf, has the dwarf phenotype. Now we do this cross, if we look at the offspring, they're all, well, I plus, I minus, heterozygotes, just like over here, but they're all dwarf. So, wow, okay, so when scientists saw this, this is amazing, what is going on here? So, and it turns out that, well, they learned, with a lot of research, is that the phenotype is determined by the paternal allele. So paternal, the allele that it's inherited from the father. So here, the paternal allele is I plus, so we're normal. And over here, the paternal allele is I minus. And I'm sure you know, but just in case, the other allele, this is the maternal. Maternal, inherited from the mom. So here, the paternal allele is I minus. And this is the one that matters, that is expressed in the mice. It doesn't matter that there's an I plus here. This is the maternal allele. It's not expressed. Only the paternal is expressed. And because this is mutant, these mice end up dwarf. And so we're going to look at why the paternal allele is the only one that's expressed in these mice here. So now it's related to DNA methylation, and I need to address something very important here. So in past videos, we said DNA methylation is typically associated with preventing transcription. So typically associated with preventing transcription. So in this example, the opposite is occurring, right? The gene that's expressed is, we're going to see, is the one that's associated with methylation. So before we get into what's going on here, I just want you to know, if you think I'm crazy and you're like, hey, what in the last videos we said DNA methylation prevents transcription usually. And I think we said usually in those previous videos. So I just want to emphasize that normally it's associated with preventing transcription, but in this case, we're going to see the opposite is occurring. So possibly the best way to take a look at this is start, let's say, with a male mouse. And now what we can do, in the past 2 minutes or 3 minutes, we talked about I plus alleles and I minus alleles. Forget about that I minus allele. We don't need it anymore. We're just talking about I plus alleles now. So in a male mouse, let's take a look at DNA holding or encoding those I alleles. So let's say this is from this one right here. This is the I plus. This is my DNA. It's beautiful, huh? I plus, this represents the I plus allele from mom. This one represents the one from dad, and it's methylated. So the cytosines are methylated here. And every cell, let me restate that. Every somatic cell, non-germline cell, in this male mouse will have this same arrangement of non-methylated on one allele, the one from mom, and methylated on the allele from dad. Why is that? So maybe you remember in the previous videos we talked about maintenance methylation. So what happens during DNA replication? So let's say this allele, I plus allele, is fully methylated. After it's replicated, it will give rise to 2 new DNA molecules that are hemimethylated, and then a DNA methylase will come in and make it fully methylated again. So this will always be methylated. This one, it doesn't start out methylated, so it never becomes methylated. It remains unmethylated. Now, I said all somatic cells. So it turns out during spermatogenesis, now early in meiosis, maybe even before meiosis really begins, so very early in meiosis, what's going to happen is the methylation marks are going to be erased from the paternal allele. So the methylation marks are erased. And it turns out that most methylated regions of the genome the methyl marks are erased early during meiosis. So now we have 2 alleles. It doesn't even matter which one's maternal, which one's paternal anymore, because neither has methylation marks. Now, as this cell proceeds through meiosis, eventually we're going to get the spermatids and the mature spermatoons, and spermatozoons. I really don't like the terminology of the different structures with respect to spermatogenesis. I'm really only interested in the 4 products, the spermatids, but the mature sperm are called spermatozoons, I guess, or spermatozoa. Anyways, each of those is going to get 1 I plus allele, and both of those will be methylated. So what happens is sometime during meiosis, these guys that were not methylated, they both become methylated. So going into meiosis, 1 the paternal allele was methylated, it was erased. So at some point during meiosis, there was no methylation on either of the I+ alleles. And then as it's going through meiosis, they become methylated again. Both alleles become methylated. Well, one becomes methylated again, and the other one becomes methylated for the first time, right? So at the end, all paternal alleles, I can't even use paternal anymore, right? Because, you know, both of these, for the next zygote, both of them are going to be considered paternal by that next zygote. So let's just say at the end of meiosis, all the I+ alleles are methylated. Okay, so that's how the paternal allele becomes methylated during spermatogenesis. Now, we should also take a look at what's going on in those female mice. So, same thing, they got two I+ alleles. One from mom, one from dad. And so the one from dad is methylated. And the one from dad will always be methylated by maintenance methylation, right? Maintenance methylation. Maintenance methylation maintains methylation in all the somatic cells of the female mice. Maintains methylation on the paternal I+ allele. So the maternal one here is never methylated. Now, same thing happens during oogenesis. Early in oogenesis, the methyl marks on the paternal allele are erased. So we have two I+ alleles, neither are methylated. And I'm just going to die, you know, one of these is going to end up as a polar body, right? Well, the products of meiosis, yeah, so we're not going to worry about that. What I just want to show you is any egg that comes out of this process, no matter which allele it gets, neither one is going to be methylated because the I+ alleles are not remethylated during oogenesis. So egg cells will not be methylated. Well, will not have an I+ allele that is methylated. Now, I guess we should take it one step further. Here's the sperm with a methylated I+ allele. Here's an egg, no methylated, oops, terrible DNA. I+ allele not methylated. They come together and we get a zygote. The maternal allele, this is the zygote, the product of fusion of the sperm with the egg. This is the zygote, maternal I+, not methylated, paternal methylated. And this is the one that's going to be expressed throughout the life of this organism here, because it's methylated. Now, scientists have worked out why the methylated allele is the expressed one in this system. So let's take a look at a quick diagram of the IGF2 locus. Remember, I was calling it I, but you know, I'll switch to IGF2 here. So here's the IGF2 gene. Let's say this is part of a chromosome. Here's the IGF2 gene. Now, let's say the gene goes this way. Upstream of the gene is a locus called ICR. Downstream is a locus called DMR. We don't need to worry about what these stand for. Now there's another gene up here which, you know, we don't really need to worry about at all, but what's interesting about it, this gene H19, is that we know there's an enhancer here, and we talked about enhancers already. This enhancer controls IGF2 expression. And one of the neat things about enhancers is they can be located rather far away from the gene. And here, the enhancer is located upstream of this gene, right here. Now, okay, this is what the locus looks like. When I say locus, I'm using it as a sort of general region. So the IGF-2 locus is sort of the general region around the IGF-2 gene. Now, when ICR, I should say, what happens here, there's another protein called CTC, and what CTC does is it binds to ICR and DMR. So let me diagram that for you. And when it also binds to itself. So if this is ICR here, this is DMR down here. When CTC binds, it causes this structure to form, this little loop here. And that's where the IGF-2 is found. So who knows? Is it really a loop? But what we don't really know. But what this symbolizes is that when CTC is bound to here and bound to here and then bound to each other, it puts this into a, let's say, transcription resistant structure. So it can't be transcribed when CTC is bound to ICR and DMR. And then here's H19, and here's the enhancer up here. However, when ICR and DMR are methylated, CTC cannot bind. So I can just put little methyl groups here. CH3, CH3. So we were saying IGF-2 has been, when it's methylated, you know, it allows transcription of the IGF-2 gene. In actuality, it's methylation of the ICR region and the DMR regions, which are right next to IGF-2. When these are methylated, CTC cannot bind to them, and therefore the enhancer can activate transcription of IGF-2 to levels that allow to levels that allow its, well, I'm stumbling over my words here, but the enhancer can control transcription and activate transcription of IGF-2. Okay, I think that makes sense, right? So this is why the paternal allele is expressed. This is why the maternal allele is not expressed. The maternal allele always has the CTC protein bound to ICR and DMR. Paternal allele never has CTC bound to ICR and DMR, and so as a result, this one, maternal allele, is never expressed, and the paternal allele is always expressed. I used a lot of always and nevers there, which is always dangerous in biology, but the important thing is we understand the concepts of how this process works. Okay, so that's it for that video. We have in the next video, one last video before our test on Thursday, and this video is the next one is going to be on how diet can influence gene expression through an epigenetic process. So diet and gene expression. So another, I think, really interesting story to tell you guys. Okay, see you in the next video.